I'm probably the one who's furthest away from the liver, being all the way in the eye, all the way at the end of the optic nerve. Briefly, I'll be talking about a little bit of background, a little bit about the effect of excess ammonia on astrocyte and neuronal signaling, some of the stuff that we've looked at. Since I only have 15 minutes, I'll also then go straight on to a little bit about future prospects. And the main technique that I've used for my thesis work, or me and my wife have actually done most of the work together. My wife was supposed to hold this lecture. She was unfortunately ill. Is two-photon imaging, and this is a two-photon image of mouse cortex, essentially cortex. So I think astrocytes are a very exciting cell type, but they were poorly understood up until the end of the 80s or beginning of the 90s because they're largely electrically inactive and signal with calcium. They're the predominant cell type in the brain, and you'll find some of the older papers saying that they outnumber neurons 10 to 1. It's most likely in cortex, at least. Of humans, the ratio is more like this. They initially got their name because they're stellate-shaped. So when Ramon Santiago Cajal stained them with silver chromate, he found that they had star shapes. This is actually not completely true. So if you patch an astrocyte, this is a human astrocyte, this is a mouse astrocyte, and inject it with a dye, you see that their actual structure is more like a bush. And in this whole bush, you can see that the soma, the cell body, which is what we often look at in cell cultures and other things when we measure things, is only a very small part of this complex structure. One of the interesting things as well is that one of the things that changes the most through evolution from simple nervous systems to more complex ones is actually the complexity of the astrocytic processes, much more so than neurons. So maybe a prerequisite for having complex, smart brains. These are some pictures from Nancy Anna Oberheim in our lab. So each astrocyte covers about 160,000 synapses in the human. And it ensheaths more than 99% of the vasculature. There's a recent EM reconstruction study showing that it's more than that even. And that's why we think that astrocytes function as a sort of go-between between the synaptic communication and the blood vessels, as you can see here, ensheathed by astrocytes. So ammonia has a lot of effects on the brain, and I think this is what this session is about, so I won't go into all of this, but it can have effects on a lot of cell types. I think we've mentioned endothelial cells. I haven't even shown leukocytes here. Astrocytes, the synapse. There hasn't been much mention of the choroid plexus, but it does have effects here as well, and I'm sure in coming years we'll see more about the effects on pericytes and other cell types. So we thought when we wanted to look at the effect of ammonia on the brain, being primarily interested in neuronal signaling, we thought we'd start with the most important part, arguably, the synapse. And since the 90s, the synapse has been redefined to include not just a presynaptic and postsynaptic neural membranes, but also an astrocytic process. This is called the tripartite synapse model. There's lots of publications on this. And the basic paradigm here has been for a long time that neurons, when they release neurotransmitters such as glutamate, they can activate the astrocyte. The astrocyte can sense this. And it was thought to sense this via metabotropic glutamate receptors primarily. This actually turns out to not be the case. So this is one example of issues with using pup cultures and pup slices, because in pups there is a high expression of MBOR1 and 5, but there's none in adults, in adult tissue. And we believe, we have some studies, 2 or 3 coming out, that show that perhaps one of the main signals that neurons release is the elevation of extracellular potassium and the small change in volume in the synapse. So here's a very simplified diagram of what we know. This synapse is upside down, just FYI, to make it simpler. So we know that ammonia enters the brain, even through an intact blood-brain barrier, although it has relatively low permeability. It can then enter astrocytes, and I believe if you expose an astrocyte to ammonia for longer than a couple of minutes, then the primary method of entry is as the ionized form. Through potassium transporters. This is similar to kidney cells. And then the reason everyone talks about astrocytes being the main target of ammonia toxicity has to do with these old studies showing that astrocytes are the only cells that express glutamine synthetase, the only enzyme that can fixate ammonia. So you'd think that they'd trap it. And some of the earliest studies by Arthur Cooper showed that most of the radiolabeled ammonia does get trapped in a small compartment of the brain. This then goes into the glutamine-glutamate cycle, which is responsible for the 2 main neurotransmitters in the brain, the main excitatory and main inhibitory neurotransmitter. When we looked at this in our lab, we just wanted to see what step might be most important for developing neuronal dysfunction, which can then lead to neurological features. And this is a very complex field, and we were interested in finding out what does actually just ammonia do. Not because if you give a mouse hepatic encephalopathy, there's a lot of other changes, bile acids, systemic inflammation. We want to know what does just ammonia do, and to do that, you have to not get too many compensatory changes. You have to also look relatively early on in the disease. It just so happens that you can't do two-photon imaging for too many hours either, so it kind of fits with that. This is just another note. We have a paper from 2012 that shows, because I know a lot of you guys who work in vivo work with anesthetized animals for regulation reasons. But anesthetics, almost all anesthetics that have been tested to date cause a paralysis of astrocyte signaling, a very, very pronounced paralysis. So just so you know that's the case. This is why we applied for approval to look at awake animals in vivo. We did stereotactic surgery, and we could then examine phenotype and look at cellular changes and electrophysiological changes in the same model and correlate the two. And this is again in adults to try and avoid the problems with that. There are some very old German studies that when they started looking at the neurophysiology that stated, we found when we looked at this that ammonia and potassium have a very similar hydrate to radius and charge. So if you start with that, start with the basics, then we thought, what might ammonia do to the potassium concentration in extracellular space? And as I mentioned at the beginning, we believe that this potassium is one of the key signals at the tripartite synapse between the neuron and the glial cell. And some of this has been looked at before in cell cultures and things, so I guess it comes as no big surprise that if you expose the mouse to an ammonia load, either parenterally or directly on the cortex, you get a brisk increase in extracellular potassium, even when you correct for the ionic interference, as you have to do the Nikolsky equation for these things. We then asked, does this increase in potassium, is it due to an impairment of transport? Because a lot of studies have said that the ammonia can cause the astrocyte to dysfunction, so maybe it's just a dysfunction of the normal transporters of the sodium potassium ATPase of the mitochondria, or is it actually just due to competition? This doesn't conclusively show it, but here we just show in a culture that sodium potassium ATPase uptake is dose-dependently inhibited by ammonium chloride. This was the same for ammonium acetate, and if you have a cell-free assay with just the sodium potassium ATPase transporter, the ammonium chloride can run the transporter just as efficiently as potassium chloride. This is the same for studies that have been done, I think, almost 5 or 10 years ago in kidney, where they showed this, and the kidney apparently uses this to flux ammonia in its normal pH regulation. So we believe it's at least in the acute phase, it's primarily because of competition. And then this issue is a bit of a thorny issue. We wondered, is the potassium increasing simply because the astrocytes are swelling, making the extracellular space smaller, and then therefore increasing the concentration? In the acute phase of isolated hyperammonemia in our model, we were unable to find any volume changes in the astrocytes. I believe there's a whole work session based on this tomorrow where I'll discuss it a little bit more. But the bottom line is we've looked at a lot of different models of diseases where you're supposed to get where you get brain edema on MRI. We've looked at hyposmotic stress, water intoxication. We looked at corticosporidic depression, where you get 50 millimolars of extracellular potassium or more. And in all these models, in vivo, the astrocytes have a remarkably robust volume regulation capacity. So they don't actually change volume as much as they do in culture or as they do in pup slices. And we didn't find that aquaporin 4 deletion was protective in this model or the model of azoxymethane, although there are some differences here in the literature. I know Mike Norenberg's group have shown that AQP4 deletion is beneficial in thioacetamide-induced HE. So we find an increased potassium. It's not due to swelling. And the potassium may serve as the main signal between the astrocytes and the neurons. So we wanted to see what does it do to astrocyte signaling. And here I have a movie. It's a little bit low resolution. I'm sorry about that. The astrocytes are loaded with the calcium indicator rod 2. And as when you get calcium activity, it becomes more yellow. This is a 50-minute baseline. The minutes are up here. As you can see, during normal baseline recording in an awake mouse, you have these sparkles of activity, but also these waves. We think it's when the locus coeruleus is activated. If we move forward to 50 minutes, and the mouse receives a dose of ammonia, an ammonia load. This is an OTC mouse in this case. And there's an initial increase in activity, but then there's a lull at the in the middle, and there's an increase afterwards. This data is a bit complex, as often with in vivo things, but you would expect if extracellular potassium increases, you'd expect calcium signaling to be stimulated, because this then stimulates the sodium potassium ATPase and different potassium buffering mechanisms. It does increase at the beginning, but the interpretation we have of this is that something else is happening here in the middle, whereby the astrocytes, the potassium stress, potassium and ammonium stress becomes more than the astrocyte can actually handle, and their signaling starts to dysfunction. It also becomes dysynchronous. Yeah. We haven't quite teased this data apart yet, but so we know that there's a lot of potassium and ammonia in the extracellular space, and the astrocytes are incapable of handling this efficiently. It's not so much as with cortical spreading depression. It's more in the range of 2 to 4 millimolar. In spreading depression, it's 50 millimolar. What effect might this have on neurons? And here there's some old studies that have looked at this as well, but more in isolated cells and things like that. Here we looked at it in vivo in awake animals. We had a paradigm to look at disinhibition, where you stimulate the whiskers of the mouse, you record in the contralateral somatosensory cortex. The second EPSP is supposed to be inhibited. It becomes disinhibited by ammonia. So we developed this model here, where the excess ammonia and potassium might act on NKCC1, which is expressed on both astrocytes and neurons, causing an accumulation of chloride inside the neuron, which then shifts the GABA reversal potential. And indeed, we found a shift in the GABA reversal potential. And when we patched individual neurons, so we were looking at neurons here, so the NKCC1 expression on astrocytes can't explain these observations. In wild types and NKCC1 knockout animals, we found that this disappeared. So it was NKCC1 mediated, not KCC2. And I think that there's at least 3 studies now showing that bumetanide can be helpful. I think there's one that's showing it might be helpful because of the effects on astrocytes. We're showing it might be helpful because of the effects on neurons. And there's one, I saw a poster, because of the effects on the endothelial cells. So here's just a summary of the effects. So just to summarize, in isolated acute hyperammonemia, ammonia itself can have effects on astrocyte neuronal interactions and signaling that are independent of astrocyte swelling, but this is in a very hyperacute model with high levels of ammonia, and the clinical relevance is a bit questionable. And I just have some queries here at the end. I just think it's important to realize what you're talking about, because people sometimes mention astrocyte swelling and brain edema synonymously. Brain edema can be interstitial, intracellular. It can be in neurons as well. So these things are not equivalent. And diffusion-weighted MRI, to my knowledge, cannot distinguish between intracellular and interstitial edema. You can distinguish vasogenic edema, but not interstitial. So simply saying that, for instance, an increased DVI signal on MRI means that astrocytes are swelling because they have GS isn't necessarily the case. HE is different in many respects that we mentioned here in this lecture. And I just think an idea for the future might be also to look at this recently described system that we worked on in our lab called the glymphatic system, whereby CSF bathes the brain, paravascular CSF, and that's how the brain washes itself, gets rid of things. And we've got a recent review coming out on this in TIMS. Yeah. So that's it. Thanks.